Hello, my name is Kayla Watts, and today I'm going to be talking about cracking the genetic code. So first off, let's talk about two things. What is the genetic code and who cracked it? Well, the genetic code is a sequence of nucleotides in DNA and RNA that determines the amino acid sequence of proteins. Now, who cracked it? Nirenberg and Mattaei was the first experiment, and Nirenberg and Mattaei was the second experiment. Now, people before all this knew what mRNA, ribosomes, and amino acids could be mixed to make proteins. They just didn't know the specific nucleotides that coded for what amino acid. So the first experiment was Nirenberg and Mattaei, and this was in 1961. And what they did was take poly-U's, which is just a ton of U's lined up, and they added this to a cell-free translation system, a test tube that contained it. And now, what is a cell-free translation system? Well, you take 20 amino acids, one radioactive labeled amino acid, and 19 unlabeled amino acids. And after you take the poly-U's and add it to the cell-free translation system, you incubate the test tube at 37 degrees Celsius and translation takes place. Now, after translation takes place, the precipitate protein's formed, and you have free amino acid in the protein. So after you have the protein formed, the protein is filtered and then checked for the radioactivity. Now, with this, it was repeated in 20 test tubes. Each test tube contained a different amino acid, and the results were, you could see what the homopolymers coded for. So you could see that AAA was coded for lysine, CCC, proline, etc., except for GGG coded for nothing. Now, why did they continue searching and doing research? Well, because they could only code for homopolymers. So only AAA, CCC, they couldn't do like GUA, for example. So that's when the second experiment happened in 1964 with Nirenberg and Mattaei. So there was new technology, so they were actually able to use tRNAs, which is a big reason why this ended up working. So they mixed three things together in their experiment. They mixed short mRNA or codons, they mixed tRNAs and amino acids, and they mixed a ribosome. So they mixed it all up, and they found unbound tRNAs and then a ribosome with an RNA and a tRNA specified by a codon. So after this, they were able to filter the solution, and so they filtered this through a nitrocellulose filter. The tRNAs paired with the ribosome-bound mRNA stuck to the filter, whereas the unbound tRNAs passed through it. So, in conclusion, for this specific experiment, mRNA with GUU added tRNAs on filter bound to valine. Therefore, GUU specifies for valine. So, many codons were determined, and they cracked the genetic code. This was very, very important to genetics. Thank you so much for listening, and have a wonderful day. Also, here are my references.